Now, let us proceed with another interesting topic, scanning electron and atomic force microscopy. So, we've learned earlier that transmission electron microscope has been exploited most widely in the examination of the internal structure of cells. In contrast, scanning electron microscope or SEM is utilized primarily to examine the surfaces of objects ranging in size from a virus to an animal head. The goal of specimen preparation for the scanning electron microscope is to produce an object that has the same shape and surface properties as the living state but is devoid of fluid as required for observing the specimen under the vacuum. Because water constitutes such a high percentage of the weight of living cells and is present in association with virtually every macromolecule, its removal can have a very destructive effect on cell structure. When cells are simply air-dried, destruction results largely from surface tension at air-water interfaces. Specimens to be examined in the scanning electron microscope are fixed, passed through a series of alcohols, and then dried by a process of critical point drying. Now, to clearly understand the difference between transmission electron microscope and scanning electron microscope, I've prepared here a diagram or an illustration. No, so we have here the difference between TEM and SEM. Now, for transmission electron microscope, the electron beam is focused by the condenser lenses to simultaneously illuminate the entire viewing field. For SEM, on the other note, electrons are accelerated as a fine beam that scans the specimen. For transmission electron microscope, electrons pass through the specimen to form the image. For the scanning electron microscope, the image is formed by electrons that are reflected back from the specimen or by the secondary electrons given off by the specimen after being struck by the primary electron beam. Now, I hope this illustration somewhat helped you to better understand the difference between TEM and SEM. Now, let us proceed with atomic force microscopy. Although it is not an electron microscope, the atomic force microscope or AFM is a high-resolution scanning instrument that is becoming increasingly important in nanotechnology and molecular biology. It operates by scanning a sharp, micro-sized tip or the probe over the surface of the specimen. Atomic force microscopy provides an image of each individual molecule, and the development of high-speed FM now allows researchers to obtain rapid sequential images of a macromolecule so that its activity can be followed over real time. So that would be all for the topic 18.3, scanning electron and atomic force microscopy. Thank you.